Hello friends, welcome to Gurum's classes. So today we'll be discussing transcription. So transcription is a very important topic. It's because transcription means it is the process. It is the first process which helps the body to make protein, because it is the process by which the code that we have in our cell, because you know very well, this is the cell. Suppose an animal cell. The animal cell has the cell membrane, and then inside we have the nucleus. So this is the cytoplasm. Fine. So inside the nucleus we have the genetic material. Well, now the genetic material contains the code that we have. Whatever we are, whatever I am right now, is because of the code. The code is continuously, the code is present in the genes, the code is present in the chromosomes. So that genes continuously keeps on making me as a male, keeps continuously promoting me to grow as a human being. So all that is present in that code. Now not only that, the various hormones, the enzymes, the digestive juices, everything is produced because of that code. And where is that code present? I told you beforehand, only that code is present inside the nucleus, and inside the nucleus also in the chromosomes. When we open the chromosome is made up of DNA. The DNA contains segment which is known as gene. Those genes, they code for the particular proteins. So the process of formation of proteins from these genes is known as central dogma or protein synthesis, and which is divided into two phases. Phase 1 is known as transcription, and then the phase 2 is known as translation. Together it is known as what? Central dogma or protein synthesis. So let us first study the first phase, that is known as transcription. Now what is transcription? So when DNA has to make the protein, it does not itself go and make the protein. It gives an order, because it says that I am the master copy. I have the code, so it gives an order to whom? It makes a servant. The servant, the one who serves for the DNA, is RNA. So from DNA, the code is taken by RNA. So from double-stranded DNA, a single-stranded RNA is formed. And when this RNA is formed, this RNA takes the code from this DNA, and this RNA is formed. Now this RNA contains the code from the DNA. So the DNA must have given the code to the RNA saying which protein has to be made. So from this double-stranded DNA, a single-stranded RNA is formed, which bears the code of the DNA, and this RNA then later on forms the protein. So in this class today we will discuss only how RNA is formed from this double-stranded DNA. So here when we talk about the process of formation of single-stranded DNA from this double-stranded DNA is known as transcription. Is known as what? Transcription. So transcription is the process of formation of single-stranded RNA from double-stranded DNA. So this is known as transcription. So in the later half, in the next class we'll study about the process of formation of protein from RNA. So from when protein is formed from RNA, it is known as translation. It is known as what? Translation. So what are we studying right now is what? Transcription. Now, as you know very well that the process of transcription, is also same like the process of replication. You know very well DNA is double helical in structure. So it is in the form of a helix. It's winded up. So beforehand it has to open up. So all those enzymes that are necessary for opening of the enzyme, like the helicase, so and then the isotopoisomerase, they are important. So they open up. Those enzymes, they open up. And when they open up, then what happens is that only when they open up, then the formation of the RNA takes place. Fine. So few things that you need to know here is the process is same as DNA replication. And see here I have made these two strands. Just imagine these two strands are now opened up. And I have already told you in the previous class also, our DNA has polarity. So this is one strand of DNA, this is the another strand of DNA. There are two strands which I have shown in parallel lines. And when we talk about the polarity, this is 3' and this is 5'. since opposite polarity are, since the DNA strands are complementary to each other, okay, so the next DNA strand here has opposite polarity. That means the 5 prime and 3 prime, they are just opposite to each other. Okay, so I have shown you here 2 DNA strands here. Fine. So from here, what is to be made? An RNA is to be made, that is single stranded. Okay, so how does the process begin? Is before beginning a process, so as you know, whenever you start anything, or whenever a project is to be started, or like say suppose in the school also, before we start the school, okay, what we do is we do the morning prayer. Okay, then after the morning prayer, okay, finishes, then the classes get started in the school, and then from 3 to from early in the morning, from 8 till 3 o'clock we do our school classes. Okay, then after the classes are over, fine, then again the bell rings, and then what we do is we do the school prayer in most of the schools, and then okay, the classes get stopped, and then the teaching gets over for the day. Okay, so that means the morning prayer, okay, signifies the start of the day. Okay, the classes, fine, then the classes takes place, okay, throughout the day, fine, and then at the last, okay, after the 8th period is over, again the bell rings, and then the evening prayer is there, or the prayer after the class is there. So that means, okay, those 2 prayers are there, in between the classes are there. So likewise the whole process is prayer, okay, classes, and then the closing prayer, starting prayer, okay, the classes, and the closing prayer. So likewise here also, okay, for the process of formation of RNA, that is transcription, okay, there is a starting point from where the DNA says from here RNA has to be formed. Okay, and then there is a point where the DNA again gives a signal saying that till here the RNA has to be formed. After that the RNA should not be formed. Okay, so a start line and the finish line, just like a marathon race. Okay, there is certain signals, so those signals are called the segments of the DNA, the portions. Okay, so here the place or the portion of the DNA from where the RNA synthesis should form, okay, that is a specific gene, a site in the gene or in the DNA. Okay, that is a special code, okay, of the nitrogenous bases, and that is known as the promoter region. So very important thing that you should know, number 1 is what? Promoter region or the promoter gene. Promoter gene is a certain specific part of the DNA, okay, which gives an order, or which signifies the place from where transcription should start. It promotes the process of transcription. So that is known as what? Promoter, okay, gene. Fine. And then the structural gene. After the promoter gene is the structural gene, which signifies the region of the segment of the DNA that has to be transcribed or has to be translated. Okay, let's not use the word translated. In fact, has to be coded. Yeah, we can use that word. Okay, to form the RNA. Fine. So here the structural genes means the total length of the DNA which has to be coded to form the RNA, or which can be coded to form the RNA, is known as what? Structural gene, the portion of the DNA. Fine. And then at last comes the terminator gene. The terminator gene means that gene which signifies that, okay, fine, the process of coding, okay, from me stops. That is why I have, okay, shown here with red color. It means stop. Okay, terminator gene signifies the coding should stop there. From there no more further coding should take place. So terminator gene is the gene which stops the process of coding of RNA. Understood? So there are 3 types of genes present, okay, in a segment of DNA, which is responsible for the formation of RNA or transcription process. Fine. So whenever RNA is formed, these 3 things are very important in a segment of DNA. That means a promoter region, a structural gene, and a terminator region or a terminator gene. That's very important. Then only an RNA will be formed, and from RNA polypeptide chain protein will be formed. Okay. So as I told you very well, that the process is as same as DNA replication. Fine. So from DNA what will be formed? RNA will be formed. So this is double stranded DNA, and from double stranded DNA what will be formed? Okay, a single strand RNA will be formed, and the enzymes necessary, okay, is DNA dependent RNA polymerase. So RNA polymerase, like whenever you make momo, what you need is the materials needed for making momo, like maida and all, or chicken momo, you need chicken stuffs, okay, like vegetable momo, then you vegetable stuffings, yes or no? So likewise, when you need to make RNA from DNA, you need is polymerase. Polymerase is an enzyme, okay, to make RNA. So unit is RNA polymerase, okay. But by looking at the DNA, so unit is an enzyme known as DNA dependent RNA polymerase. Fine. So in case of eukaryotes, that means us, in case of the higher organisms or the eukaryotes, okay, what we need is, okay, for the formation of RNA polymerase is needed, okay. And in case of eukaryotes, there are three types of RNA, that is rRNA, mRNA and tRNA. rRNA means ribosomal RNA, which later on forms the ribosome. You know very well, okay, what is a ribosome? Okay, it is the kitchen which helps to synthesize protein, and ribosome is present even in prokaryotes also, okay. mRNA, okay, so sorry, okay, mRNA means messenger RNA, okay, and messenger RNA means the RNA which takes the messages, okay, like we have is WhatsApp, okay, or any other applications, okay, and it helps to carry messages. So likewise, okay, this RNA carries messages, okay. Later on we'll come to know what do we mean by that, messages. And tRNA means the transfer RNA, okay. It helps, or it acts as a postman or a courier boy, okay, or pizza delivery boy, okay. Later on we'll come to know in the next video. I'll explain you in detail. So let us remember rRNA, small r capital RNA means ribosomal RNA, mRNA means messenger RNA, and tRNA means transfer RNA. These three RNA are formed, okay, with the help of an enzyme which is known as RNA polymerase. And okay, for RNA polymerase enzyme 1 is needed, another RNA polymerase 2 forms mRNA, and RNA polymerase 3 forms tRNA in case of eukaryotes. But in case of prokaryotes, if you see, okay, then these three different types of RNAs, that is RNA, mRNA and tRNA, are formed with the help of a simple type, that is only one type of polymerase is responsible for forming all these three different types of RNAs, okay. So now if we look into the process of this transcription, then here I have shown you, okay, a double helical structure, okay, in the form of DNA double helical structure in the form of parallel lines with the polarity 3' to 5', and the opposite polarity 5' to 3', and you know very well this is the process which has to be transcribed to form RNA, and which contains the three units, that is the promoter gene, the structural gene, and the terminator gene. This is known as transcription unit. Okay, you can note it down. What is a transcription unit? A unit, okay, it is a segment of DNA which contains these three genes. Fine. This is the promoter gene, structural gene, and the terminator gene. Fine. And now you know very well, okay, this is the DNA strand, this also is the DNA strand, and the difference between two DNA strands is only one thing, that is the position of the nitrogenous bases. The position of the nitrogenous bases. So here, okay, here I have written, okay, here, suppose if you focus upon this first strand here, okay, from 3' to 5', okay, then I have written certain nitrogenous bases here in random, okay, and you know very well this is DNA, so that is why DNA contains, okay, guanine, thymine, okay, cytosine and adenine. Fine, uracil is absent. So what happens is that when the transcription starts taking place, the DNA has opened up, polymerase has already started, okay, adding up the new RNA strand. Now the main thing here is when RNA is formed, okay, the RNA has to take the code from the DNA. The RNA has to take the code from the DNA. Now the RNA questions, okay, from which strand should I take the code? Because there are two strands, either this strand or either this strand, okay. Then the answer is the RNA which is formed new always starts from, you know very well, the polarity, that is 5' to 3', Okay, you know very well the formation always starts taking place from what? 5 prime to 3 prime. And when this polarity has to be maintained, then that strand has to be taken place which has the opposite polarity. So here I have already written a polarity of 3 prime to 5 prime, and the opposite is 5 prime to 3 prime. I cannot take this as the strand from where the coding has to be taken place. Okay, if I take, then again it will be 5 prime to 3 prime. Okay, and then the polarity will be same. So that is why the upper the strand will be taken as the coding strand. Okay, so that is why from where the code will be taken place. Later on I'll come to I'll tell you which is the coding strand. So the upper strand, okay, will the code will be taken from the upper strand. So here I have shown you. So this is the upper strand, this is the lower strand, and now the DNA polymerase has started, okay, forming a new RNA, which I have shown you in the form of, okay, red colored DNA RNA strand. Okay, and here the promoter region has already started, and then terminator region is here from where the RNA formation will stop. So that means this is the structural gene from, okay, this is the structural gene within which the formation of the RNA take place. And here I have even written the nitrogenous bases also. So here is GTTC, ATC, okay, these all are the nitrogenous bases, and opposite to them was here, or they all are here. If you see G, GC, okay, T, A, T, A. If cytosine is there, guanine is there. So these are opposite. But when RNA is formed from, okay, this strand, or it should be formed from this strand only because the polarity is opposite. Okay, so when the RNA will be formed from, okay, looking at this strand, then G, this is the, okay, the strand of the RNA. G will have opposite polarity is cytosine. Thymine will have adenine, cytosine. Okay, adenine should have thymine, but you know very well that RNA does not have thymine. Instead of that, it has what? Uracil. So I have written uracil. Okay, I have written what? Uracil. Thymine again contains adenine. Okay, these are the opposite, okay, nitrogenous bases. So that means here this new RNA is formed by looking at the codes which are present on the upper strand. Okay, I'm just saying the word upper strand. You shouldn't be using that word upper strand. The strand which contains the opposite polarity. So this is known as the template strand. This is known as what? The template strand. Okay, so template strand. So that means template strand is the strand, okay, by looking at which the new RNA strand is formed, is known as what? Template strand. You know, even in school time also we used to use these stencils. We used to have is a ruler or a big stencil where we used to have alphabets A, B and C. So that is the template. Yes or no? By looking at them, or we draw lines or we make alphabets. So likewise, okay, this is the template. Okay, and then the opposite strand. If you see the code of the opposite strand, it matches with the newly formed RNA. So here, okay, if you see this strand and if you see this strand, only the difference is the presence or the absence of thymine. Okay, so that is why this strand is known as what? Coding strand. Okay, this strand is known as coding strand. Coding strand. So that means there are two strands of DNA, of which only one strand, okay, will be taken, okay, into consideration by the RNA, and the code from that strand will be taken, and that strand is known as template strand, and the non-coding strand is known as the coding strand. Why is it called coding strand? Because actually it matches with the code of the RNA. And then after the DNA has opened up, and once the RNA has formed, the opened DNA again recoils, and then, okay, the original status of the DNA is again maintained, and here I have shown you the newly formed RNA is formed here. Okay, but in the next class, in the next video, I'll explain you this newly formed RNA also, has many impurities. You know very well, when gold is mined from its ore, it contains many impurities, okay, and again it has to be purified. So likewise, this newly formed RNA also contains many regions, okay, which actually are not needed. Fine. So afterwards, once it gets filtered, okay, purified, then later on it comes out of the nuclear membrane. So suppose this is the nuclear membrane, okay, suppose this is the nuclear membrane. Nuclear membrane contains many nuclear pores, fine, and then it comes out, this newly formed RNA, then this newly formed RNA comes out of the nuclear pore, okay, from the nuclear pore, okay. But this RNA, before coming out of the nuclear pore, has to undergo, okay, one more stage of purification. Fine, that I'll explain you in the next class. So in gist, if you want to know what is a gene is a segment of DNA, okay, which acts as a hereditary unit. Simple. But if the question comes, what is a cistron? A cistron is that segment of the DNA, okay, which actually codes for a protein, okay, you know, that codes for a protein or a polypeptide chain. Cistron is that segment of the gene actually which codes for a polypeptide chain or a protein. So cistron, okay, actually cistron means polypeptide chain. It is related to polypeptide chain. Polypeptide chain of what? Means it to form protein. Polypeptide chain of amino acid to form what? Protein, okay. And monocistronic and polycistronic, you know, when we talk about monocistronic, in eukaryotes we have is one cistron, okay, coding for one protein, fine. But in case of the lower organisms like prokaryotes, there are many cistrons, that means many segment of DNA, okay. When we talk about in general term, many segment of DNA, actually these all are cistrons. Together they form RNA. Then these all RNA, 3-4 RNAs, they club together to form one protein, okay, fine. So you know, it's just like singles game and a team game, okay. Volleyball is a team game, okay, whereby 6 members they play for one trophy, okay, or for one goal, okay. And when you talk about badminton single, one person plays, okay, one game against one person, okay. So that is one single game, yes or no, okay. So one person codes for one protein, okay. So here 6 persons they are coding for one protein. So this is monocistronic, and this is what? Polycistronic. And polycistronic normally takes place in case of prokaryotes. So friends, I hope you all have understood very well, okay. So this much for today. In the next video we'll discuss about translation, okay. So thank you very much.